Hello, I'm Byron Cheatham with CytoViva, and today we wanted to just show you really quickly how 3D imaging of nanoparticles or nanomaterials in cells and tissue work with the CytoViva 3D microscopy system. And here we want to show two images on the left and the center of fluorescently labeled cells, where the cytoplasmic area is green, the nucleus of the cell is blue, and the red dots you see are nanoparticles that have been internalized by the cell structure, or in the case of the image on the left, are sometimes internalized by the cell structure, but also on the outside area of the cell structure. And we're using a algorithm called point of brightest voxel that allows us to see unlabeled nanoparticles, label-free nanoparticles, interacting in this case with fluorescently labeled cell elements so that you can see in the X, Y, and Z where the nanoparticles are located. On the right, we're showing some lung tissue, and it's lung tissue where carbon nanotubes, multi-wall carbon nanotubes, have been internalized in the lung tissue area. Here, the lung tissue is not fluorescently labeled. The deconvolution is not quite as crisp as it is in the fluorescently labeled cell structure, but clearly you can see the red pseudo-colored carbon nanotube that's internalized in this tissue structure, this lung tissue structure that you see, which is shown, it looks red here in this image coming around. So these are three videos which demonstrate the ability to see nanoparticles in the X, Y, and Z in 3D optical imaging. And of course, with CytoViva, you can capture a hyperspectral image of any of the areas of 3D stacks of these images to be able to confirm spectrally nanoparticle presence in these cell and tissue areas. Thank you very much.